Today, I have Professor Monte Winslow with me. And Dr. Winslow grew up in Canada and received his undergraduate degree from the University of Victoria in Canada. He did his PhD studies in immunology, working with Dr. Jerry Crabtree at Stanford University, before pursuing his postdoctoral training with Dr. Tyler Jacks at MIT. Dr. Winslow is currently an Associate Professor of Genetics and Pathology at Stanford University. So Dr. Winslow's research focuses on understanding the mechanisms that drive cancer development, progression, metastasis, and therapy response. He's developed in vivo mouse models that enable the isolation and genomic characterization of cancer cells and use these models to contribute to our understanding of cancer initiation, growth, and progression. These studies have focused on uncovering the molecular forces that contribute to the development and progression of the three most metastatic and deadly cancer types: lung adenocarcinoma, small cell lung cancer, and pancreatic cancer. He's also contributed to the development of novel cancer modeling approaches that enable rapid and quantitative analyses of the genetic drivers of carcinogenesis. His laboratory has generated methods for somatic CRISPR-Cas9-mediated genome editing and used these models to introduce both gain and loss of function alterations in somatic cells in vivo. By incorporating increasingly quantitative methods and powerful in vivo methods, Dr. Winslow's work in uncovering general rules that govern tumor isolation, growth, and progression, Dr. Winslow's work is uncovering general rules that govern tumor isolation, growth, and progression across diverse tumor genotypes. So thank you for being here. And now I'll pass it over to Dr. Winslow.
All right. Thank you, Jan, for that introduction. And thank you, everyone who's joined this webinar. What I want to do today is really give sort of a broad introduction of how we and others have been using CRISPR genome editing to really understand cancer better. So what I want to do is really three things today. I want to introduce cancer genetics to you. I want to introduce genome editing. I'm going to then talk about some applications of how genome editing has contributed to our better understanding of how cancer works. And then I'll kind of end with some future directions. Where do I see this going? And some sort of simple take-home messages from this webinar today. So first, we'll talk about sort of cancer genetics and genome editing. So despite there being other huge global health problems right now, cancer remains a tremendously high in incidence and drives a lot of mortality around the world. This is sort of just showing the metrics of the number of people who get different cancer types and die from different cancer types worldwide each year, with lung, colon cancer, stomach, liver, breast cancer, pancreatic cancer is right kind of coming up next. So cancer remains a kind of a formidable disease around the world. But I want to show this next plot because despite the prevalence of cancer, in the United States and the world, we've actually made pretty good progress over the last 50 years. So what this chart is showing you is on the bottom axis is essentially, well, let's look at the left-hand axis first. This is a metric that sometimes people think about, which is the 10-year survival rate. If a patient is diagnosed with a certain cancer type, what's the chance they're going to be alive 10 years later? And you can see that different cancers have sort of different 10-year survivals. But as I was going to say, the important thing is on this axis on the bottom, which is like how much has that percent 10-year survival changed in the last 50 years? So for example, if you look sort of at the top right, you'll see prostate cancer. There's more than a 50% increase in how long people will survive with prostate cancer. What percent of people will survive 10 years? So for many of these cancers that are moving to the right, there's been great advances in treatment. But there is some that remain down, at the bottom left: lung cancer, pancreas cancer. These are still extremely hard to treat and where I think we need really new advances. So cancer is fundamentally a genetic disease. And what this means is that within the genome of the cells, of the normal cells that become cancer, there are alterations that occur, and it's those alterations that really initiate cancer and drive cancer progression. And these different type of alterations that can occur in the genome, they might be mutations. This means that there's single, letters in the genome, we call them nucleotides, that get changed. There can be structural alterations. There can be whole big pieces of chromosomes that get stuck to each other or flip around. And there can also be extra numbers of chromosomes. So these, we get 3 copies of certain chromosomes, and for some reason, this helps drive cancer. So regardless of what these alterations are, there's really 3 outcomes that you can get. The alteration, the genomic change, can either essentially inactivate the gene that it occurs in. And the reason that this can cause cancer is maybe these are genes that normally stop cells from growing. Now, if you lose that gene, they might grow more. Those alterations can increase the activity of that protein. Usually we call these oncogenes. So you get a mutation and it turns something on that really drives the cell to now become cancer. In rare cases, these alterations can completely alter the function of a gene. So there's a relatively simple relationship here. Genomic alterations can drive these changes in your normal cells that can then lead to cancer. But despite the sort of simplicity of that, there's many complexities. One is that many tumors have lots of alterations in them, up to tens of thousands of these alterations in their genome. Some of them might be important for driving cancer. People call those driver mutations, while others might not actually have any effect. They just happen to have gotten mutated along the way, and they don't do anything. People call those passengers. But in many cases, it's not clear which alterations in different genes are drivers or passengers. It's also the case that there are many different genomic alterations in different tumors. So tumors from no 2 patients will be identical. So there's a lot of complexity in trying to understand which of those alterations might be functionally important, might really be important for driving cancer. And then not only are these tumors different, but there's different combinations of genes. Maybe one patient has these 5 genes, and another patient has 2 of those, but also 3 or 5 others. So there's a lot of complexity there in trying to understand how these alterations in the genome of cancer cooperate to drive the way that cancer acts. So as you can imagine from these sort of understanding of the genetics of human cancer, that new tools that allow us to purposefully alter the genome, to ask questions of, if I create that mutation that sometimes occurs in cancer, does it cause cancer? Does it help cancer grow? These are the type of questions that these new tools can really help us address. So the tool we're going to talk about today is CRISPR genome editing, and the example we use is CRISPR-Cas9. It's it was one of the first ones discovered, and is sort of the prototype for how this works. These are actually natural systems that occur in bacteria that now we can use to better understand genetics. The way these work in bacteria is actually you can see on the left, there's these Cas genes. These are CRISPR-associated genes. These are proteins that are encoded in bacteria naturally. And what these proteins do is they bind to a small RNA molecule shown over here called a guide RNA. And when that protein and that RNA molecule come together, then it can actually help protect the bacteria from these phage. And phage are essentially virus that infect bacteria. So this is a way that the bacteria can defend itself essentially against these virus. But what became clear is that when you have the ability to express a protein, one of these Cas proteins, and this small RNA called a guide RNA, then if you can cut DNA in the bacteria, yes, it's a phage, but if you could use it in mammalian cells, you could potentially cut the DNA in a cell and do different things with it, maybe create mutations that we can then understand how they function. So this is what people have done. It ends up that there's really only 2 components that you need for this CRISPR-Cas9 system. You need this Cas9 protein, and you need this small RNA called a guide RNA. Cas9 is a type of protein called the nuclease. In this case, it means it can cut DNA. And the SgRNA is this RNA molecule that can target Cas9 to anywhere in the genome. This RNA molecule is actually the part that binds to the DNA and specifies where it will bind. And it's what we call programmable. You can make it have any sequence you want. And when you make that guide RNA have a certain sequence, it'll go to a certain region of the genome, and then Cas9 will cut the DNA at that location. And the reason that's useful is when you create a double-strand break in the DNA, it then undergoes these natural repair process, just like our DNA often does. If it gets broken, it can repair. But that repair is not always perfect. So you can get these small insertions and deletions that then essentially break that gene. So we now have the ability to express these two components, Cas9 and a guide RNA, create a double-strand break anywhere in the genome that can be repaired, and in some cases, it will inactivate that gene. So you can see now that we have the ability to create these alterations in the genome, much like those that naturally occur in human cancer. And I'll tell you how we've been using that to better understand how essentially cancer forms are one of the driving forces. So one key thing here that you'll see in all of my examples is that you need a guide RNA, you need the Cas9 protein, you need to somehow get that into the cells, and then we can look for the effects, and we can learn about what these genes we're inactivating actually do. And also, as you've probably seen, I've laid out a very complex group of diseases, all these different cancer types, and a genome editing tool that allows us to start investigating how those genes function. So this has really been sort of a perfect match in the development of a tool and the ability to investigate a complex disease. So the next thing I want to do is talk about some applications of genome editing that have really accelerated our ability to understand, I think, two very important aspects of cancer. So the first thing that I want to talk about is not so much trying to say, hey, these genes are broken in cancer and we want to break them and see what they do, but rather present how people have been using genome editing to understand what we often call cancer dependencies. And this is saying, what do cancer cells really need? And if you could, for example, inactivate a bunch of genes, you might learn what cancer cells depend on, and then that might be a route to understand how to have drugs that can better kill cancer cells. I'll show you a few examples. And then the second main thing I want to tell you about is really altering genes of interest in cells to try to understand which of those genes drive cancer growth and different phenotypes, different ways those cells act. So first, let's talk about uncovering cancer dependencies. So what I laid out a few slides ago was the idea that if you can get a guide RNA targeting a certain gene and Cas9 into a cell, you can then inactivate that gene and you can learn about what it does. So in a very simple way, you could imagine taking a cancer cell line. These are cells from human, often tumors that we can now grow in dishes in the lab. And you could take that cell line. If you express a guide RNA targeting a certain gene in Cas9, you can get a cell line that is essentially identical to the initial one, except that it lacks maybe one gene that you think might be important. And then we can look and see if there's any effect on those cells. And what could the effect be? It could be that the cells all die. That seems like an important gene for cancer cells. It could be that the cells grow slower. They could respond different to therapy. It could really be anything that, in the lab here, we can think about looking at and ask questions about how does that one gene function? And this is really the basis of a lot of what we understand about cancer, a lot of what we understand about biology is saying, how do we, inactivate a gene and then see what happens? You might imagine saying, well, if you can delete one gene, maybe you can delete a lot of genes all at the same time in different cells, and you can learn about what they all do. This has been really a tremendous application of CRISPR-Cas9 genome editing, saying, in this case, let's take a cancer cell line and, we'll take tens of millions of those cells and let's get it so that every cell has a different guide RNA expressing it. All these cells will inactivate different genes. We can then grow them, for a certain amount of time and just ask the question of, what's the effect? And in this case, the effect could say, if we inactivate some of these genes, maybe this library that we express actually inactivates all the genes in the genome, but individual genes in different cells. If we grow them for some time, some of those cells will grow less well. And there's ways to understand that at the end of the experiment to say, oh, here's the 100 genes that really hurt these cancer cells. Oh, here's some genes that maybe help them grow. So these are the sort of applications where you can start looking more broadly at how different classes of genes impact cancer growth or different ways that they behave using these sort of cell line studies. And this has been a huge application. I want to show one exact example from one of my colleagues here, Mike Bassick, in the genetics department at Stanford, where they took this one step further and said, well, we don't want to just inactivate one gene in these cancer cells, and we don't want to inactivate a bunch of individual genes. They did an experiment to say if we delete 2 genes in every cancer cell, these cells grown in dishes in the lab, what will the outcome be? And the important thing here is that which genes did they try to inactivate as, combinations, as pairs? And it ends up that they decided to target genes that are potential drug targets. So they have guide RNAs that target these potential drug targets, and they made pairwise combinations of all these different guide RNAs to target essentially pairs of drug targets. This is the layout of their we call them screens that they did, where they have the one guide RNA targeting a drug target, another guide RNA targeting another drug target, and they made a huge number, 21,000 different combinations of these. They can put them into this cancer cell line, and then all the pairs are represented. They grow them for some time, just like I said before, and then perhaps there's some pairs that now get lost from the sort of population of cancer cells, suggesting that inactivating those 2 genes really hurts the cancer cells. This is a plot from their paper, but the important part to look at is really down here. These are the pairs of genes that when you inactivate them really hurt this cancer cell line. And I want to highlight one, which I'm going to show a little bit on the next slide, but it's this BCL2L1 and MCL1. These are important proteins that normally keep the cell alive, but it appears that if you inactivate both of them here, you really have a huge impact on probably killing these cancer cells. So the reason to do this whole experiment was there's a certain logic here that says that if you can delete pairs of genes using CRISPR, remember, this is entirely impossible without these sort of genome editing tools, right? If you can delete pairs of genes, maybe it tells you about pairs of drugs that you should be using, because you identified those genes and what they control as being important in this genetic sort of screen that Mike Bassick's lab did. So then the next experiment they did was to say, well, we now know which 2 genes are important. So what if we use 2 drugs that also inhibit essentially the proteins that those genes make? And here's their result. This is the percent of cells that are dead. That's what the axis is showing. And if you have no drug, there's very few dead cells. If you have a drug that inhibits one of these proteins, you get a little bit of death, the other one a little bit of cancer cell death. But if you inhibit both of them, all of a sudden you almost entirely kill the cancer cells. So by doing this pairwise, genome editing screen, they were able to find combinations of, in this case, drugs that might be effective against this cancer type. So these are some, I think, kind of nice examples of how people have used genome editing, in very broad scale, right? 20,000 genes, 20,000 combinations to really try to understand what do cancer cells depend on. There's been huge efforts across, hundreds of different cancer cell lines from different cancer types to understand these sort of dependencies, as it might really help the development of novel therapies, really understanding how to hurt cancer cells. So the next thing I want to talk about is really using CRISPR-Cas9 genome editing to understand the driving forces of cancer. So yes, cancers have many mutations, but which of these are actually important in driving cancer? So one thing that I would like to point out is that cancer is really a complex process that occurs within sort of the environment in which the tumor is growing. So shown here on the left is a individual normal cell, and this is what cancer comes from. And, there can be certain genomic alterations, these changes that occur in those initial cells that drive the initial sort of expansion of those cells, and maybe they're not behaving quite like they're supposed to. And then other genomic alterations can occur over time, more of these mutations, and then the tumor gains a greater ability to grow. There's also a lot of interactions with other cells within the sort of microenvironment that tumor is. And this is one reason why we and others have really focused on trying to understand cancer growth within the in vivo context, right? Within the context of an animal that is growing these tumors, we think that some of these other interactions are really critical for understanding how cancer develops. Remember, I sort of told you that for at least CRISPR-Cas9 genome editing, all you need is to be able to get the guide RNA in the cell, Cas9 in the cell, and you need some sort of delivery method to get those two things in there. So you can imagine being able to deliver those to, again, that single normal cell, in our case, within mouse models of human cancer. And if you can get those into that single cell, you can then ask the question, okay, does that genomic alteration that you just created now drive early expansion of those cells, drive their bad behavior, allow them to grow particularly quickly, which would indicate that that gene is important in cancer. So we started doing this many years ago, and the first cancer type that we modeled using CRISPR-Cas9 genome editing within these mouse models of human cancer is actually pancreas cancer. And the way we did this is we had the Cas9 engineered within the mouse. So essentially every cell in the mouse has the ability to express Cas9. And then we could deliver the guide RNA component, this programmable RNA, on a viral vector. We use these very frequently. You can engineer these viruses to do what you want, to just deliver, in this case, this guide RNA, and one other important component here. And then we were able to deliver this virus into the pancreas of one of these mice and start asking questions about how these certain genes may or may not contribute to pancreas cancer growth. So we actually did this all in a model for the aficionados where all the cells will express an oncogene called KRAS, and then we use CRISPR to alter other genes, ask if they normally suppress cancer growth. We also engineer these mice in a way where all the little tumors will glow red. So in this case, this is a pancreas from a mouse where all these small red regions are these oncogenic KRAS-driven, very, very small pancreas tumors. But if we use CRISPR-Cas9 genome editing to inactivate this gene that my lab cares about called LKB1, it's a potential tumor suppressor. But what we found in the pancreas is that if you delete that gene, now rather than getting these really tiny pancreas tumors, you get these huge pancreas tumors. This is clearly a gene that is incredibly important in pancreas cancer. When you delete it, you get bigger tumors. We can look at this a number of different ways. This is actually a section of a pancreas. This is pretty much what a normal pancreas looks like, yours and mine. This is what it looks like. But now if you delete this gene using CRISPR-Cas9 genome editing in those cells within the pancreas, or this is not a cell line we're transplanting, we're actually genetically altering single cells within the pancreas of this animal. Now we get something that looks very different. And in fact, this looks like some sort of variation of human pancreas cancer. The key feature here is that what CRISPR-Cas9 genome editing allowed us to do is assess the importance of this gene very quickly. There are other ways to do this. They would have taken us 3-4 years to do. But in this case, we could do it in a matter of months. So that, for us, is a huge advance. That's a huge advance for what our lab is able to do and other labs are able to do as far as understanding how these genes impact cancer growing in vivo. But I kind of started this talk by telling you that cancer was very complex. Many different genes are altered. Many different combinations of genes are altered. And then I just showed an example where we went back and we deleted one gene and we celebrated and we were happy. But how do we now start thinking about using genome editing to understand more about the complexity? So what I told you is certainly true. If this is sort of a textbook model of cancer where there's a single normal cell that then gets an oncogene and it grows, and these tumor suppressor genes, these genes that normally suppress cancer growth, are getting deleted. We can just look at human data and we can say, well, lots of human cancers have been sequenced. In this case, I'm going to talk about lung cancer. And you can look at the frequency, what % of human tumors have inactivation of certain genes. And here's the ones we're going to talk about. It's sort of a list of 11 genes. And they're mutated at different frequencies in human cancer. And some of them are already genes that we know are tumor suppressors, and some of them are not. And so we decided to try to develop a system where we could inactivate all 11 of these genes in different tumors, but a way where we could understand which of them are important for driving cancer. So what I sort of showed you in the pancreas, we've also been able to do in the lung, and that would be to say, take a viral vector. Remember, this is how we're delivering the guide RNAs to inactivate this gene LKB1 that we like so much. If you deliver that to the lung of a mouse, you can generate lung tumors if it's a context where it also has this oncogenic KRAS, and that's fine. But what we decided to do was say, well, what if we made a pool of these lentiviral vectors, these viral vectors, and in each one we put a guide RNA targeting a different tumor suppressor. Then if we delivered it to mice, we can actually develop, in this case, 11 different flavors of tumors. We call them genotypes because they have different genes mutated. 11 different genotypes of tumors in an individual mouse. Then maybe we could just, from one experiment, assess how all, for example, 11 of these genes might function. So that's exactly what we were able to do. We can deliver this pool of vectors to the mouse lung. We can generate all the different genotypes. Each initial cell really only gets one of these virus, so every tumor only deletes one of these genes. And then we developed methods based on high throughput sequencing to really quantify the size of every single tumor in the mouse. Maybe they get 1,000. We can quantify them all. We can understand how many cancer cells they have, and we can know which of these 11 genes was inactivated in each tumor. We could take that information and then really try to understand the impact of these genes. But even before we did that, we knew that some of the genes in here were really important in driving cancer. This is actually a lung lobe from a mouse, and this mouse does not have Cas9. Remember, when there's no Cas9, none of those genes will be inactivated. And when there's oncogenic KRAS, you get some small little red tumors I hope you can see here. But when the mice do have Cas9, and all those tumor suppressors are being deleted in individual tumors, now we saw many more large tumors. Certainly, some of these genes that we inactivated are really important in normally slowing down growth, and when we get rid of them, the tumors grow faster. So we analyze this in many ways, but this is a sort of simple way to show it, where here are all the genes we inactivated, and if they're dark blue, it means when we deleted them, the tumors grew really big. The ones that are lighter blue, when we deleted them, the tumors grew a little bit faster, and actually these white ones, when we deleted or inactivated those genes, it had no impact on growth. So that was also interesting to us. So we could make some conclusions. One is that we actually identified novel genes. We identified genes that were not known before to suppress cancer. And in this sort of pooled setting, we could find those genes. It was interesting to us that some of these genes were actually not important, at least in cancer, how we had modeled it. And I would say that the effect of inactivating different genes, like whether they had big effect or smaller effect, it didn't correlate with how frequently they're mutated in human cancer. And the reason we think this is important is there may be genes that are altered in a small percent of human patients. And in fact, those genes are incredibly important in those individual tumors. So we need these type of approaches where we can look at large numbers of genes to really start looking at those that are rare in human tumors, but might still be very important. But I also told you that, remember, it's not in human cancer, it's not a single gene gets mutated. There's going to be combinations of genes that get mutated. So in human lung adenocarcinoma, this is the subtype that we're looking at. I showed you this before. Here's the frequency at which these genes are mutated. And these are the ones that I just showed you the impact of. But in fact, if you say, wow, there's this gene P53, it's incredibly important. See here, it's mutated in almost half of lung adenocarcinoma. So if you look at human tumors that have P53 mutations, but they also have mutations in all these other genes. So there's pairwise mutations in these potential tumor suppressors. And it's the same if we look at another subset of human lung cancer, those that mutate this gene called LKB1. They also have mutations in all these genes. So this is a sort of glimpse of this complexity I was talking about, where there's pairs of genes that are inactivated. So we did another experiment where instead of deleting one gene, we deleted the pairs and essentially created this 3x11 matrix that I'm showing you here, where we can then begin to understand when you delete combinations of genes, what happens. Remember, we don't want to delete combinations of genes just for fun. We want to understand when there's a human tumor that has that pair of genes inactivated, is that in fact working to drive the growth of the tumor? And I think you can see that from the sort of blue coloring, that in some cases when you delete pairs of genes, the tumors grow much better than if they only had one gene inactivated. So it ends up that this comes to a conclusion where we say the effect of inactivating these genes, how genes drive cancer, it's really context dependent. It really depends on what other genes have already been altered in the tumor. And this is something that wasn't really previously solidly established. So we've gone on with these type of methods, now to delete 50 putative tumor suppressors and start looking at not only do they drive growth, which is kind of what I was showing you before, that's in the middle, but, do any of them drive tumor initiation, how well a cell will go from being a normal cell to being a cancer. And then there's some other features of cancer we've been able to look at, but we're starting to develop more complex maps like this, where some of these genes, like you see this one in the top right called P10, it appears to be very important in suppressing cancer initiation and growth. So when you get rid of it, there's more tumors and they grow faster. And other genes here have very different phenotypes, different effects of deleting these genes. So this is really what we get excited about, because now we can start looking at large numbers of genes. We can really understand what they do in cancer, growing in this case in these mouse models. And the important thing to not forget is the only reason we can do this is because of the development and discovery of CRISPR genome editing, right? This really easy way to inactivate genes by just using Cas9 and this guide RNA to target them, create DNA breaks, and ultimately inactivate the genes. So I'd like to just talk about one last thing, which I think is tremendously important. And this is the question of when you have a population of patients, we actually now have widespread cancer genotyping, which means that tumors from many patients are being sequenced to find out which genes are altered. So for many, for example, lung cancer patients, they come to the clinic and we ultimately understand all the genes that are mutated. There's also a lot of drugs, as shown here on the right. But one fundamental question that is still extremely hard to tackle is, which drugs should which patients get? These patients have different genes altered, and we have a lot of different drugs, but, which subset of patients should get the red drug and which should get the other drug? So we've tried to use our in vivo approaches to begin to address this question. I told you that we can take this library of viral vectors and we can initiate tumors in mice that, in this case, are essentially 12 different genotypes, 12 different flavors of tumors. So we did an experiment where we took groups of mice with all those different tumors, and we treated them with different therapies. And then we did our same approach to look at the size of every tumor, and this allows us now to start asking questions about whether the genes that are mutated in the tumor either make them more sensitive to certain drugs, that would be good, or more resistant to certain drugs. Well, that would also be good to know, because it would say that if a patient, for example, has this gene mutated, they should probably not be treated with that drug. The genotype of their tumor will dictate the response, and that looks like maybe a genotype that won't respond. So what we were able to do was create sort of a map like this, where all the different genes that we inactivated are shown on the left-hand side, and the different drugs are shown on the bottom. And I'm going to show you this sort of heat map, but what this really shows is that any square that's red means that tumors that had that gene inactivated were resistant to the drug. So, for example, you can see some of the dark red ones here, right? That would mean that if you inactivate that gene, the tumors really don't respond. And then the blue squares mean that if you have a tumor that has inactivated that gene, they're in fact very sensitive to that drug. So that would be good to know. And the interesting thing for us here was not necessarily the individual interactions between the genes and the drugs, but that there were so many interactions, right? So these genes that we could inactivate really drive drug responses. And this suggests that the genotype of the tumor, which of these genes are altered, might be a major determinant of how well patients' tumors respond to different therapies. And we would love to think that, these type of findings can help with precision medicine, right? Understanding how the genotype of the tumor might dictate and drive which therapy they'll respond best. So I'd like to end with some sort of future directions where I see this going, at least in our line of work, and then some take-home messages. So, what aspects of cancer development, do we think are poorly understood, and where these genome editing tools could help develop new understanding? So this is sort of a drawing that some of these people do of cancer, where at the top is the cell, the normal cell that the cancer came from, and then as it goes down, those the sort of plus signs are new mutations that occur. And then sometimes there'll be, sort of different, they call them subclones, but different pieces of the tumor that are different from one another, and that's sort of these different colors. So you can think about going from a single normal cell all the way to the bottom where you have a very heterogeneous tumor. So what do we still need to be able to do better to understand cancer? As you can see with these plus signs, these mutations that are occurring in the cancer over time as it kind of goes down the slide, it would be great to be able to control when alterations occur. So we generate one mutation at the beginning, and then at later time points we can introduce others. That would be really important in understanding sort of cancer evolution. Human cancer is still more complex than what we're modeling within the mouse. On one level, the mouse that we use, these type of models, are actually very good. We can understand what genes do. But it might be nice to have systems develop even more complex alterations like those that occur in human cancer. I talked a lot today about, creating alterations in DNA, right? These mutations or, these chromosomal changes. But there's a whole nother layer of complexity, which is called epigenetics. This is where genes get changed not by having mutations, but by other elements that control their expression. There's ways to use genome editing to understand epigenetics. These tumors, as you can see here, there's a lot of different colors. There's a lot of complexity within an individual tumor. That's something that we would love to be able to understand the meaning of. Maybe the different colored clones here sort of helping each other. That would be interesting. And then I think there's still a huge need to better understand how the genotype, right, how the genes that are mutated actually dictate therapeutic responses to better understand sort of precision medicine type approaches. So with that, I'll have my last slide here of take-home messages. One is that cancer is really a complex genetic disease. There's lots of genes that are altered, and they drive the way cancer behaves. And this is important for us to understand. Second is that CRISPR-based genome editing really is accelerating our ability to investigate many different aspects of cancer development, from cancer initiation, growth, metastasis, drug response. Every step along the way is now kind of benefiting from being able to do these types of experiments. And finally, I think that these type of advances, remember, we went from being able to inactivate one gene to inactivate, in cells in culture, 20,000 genes, and in the mouse, 50 genes. These are huge advances, and I would love to think that some of these approaches are going to be important, not only for understanding cancer, but thinking about how to treat it better. So thank you very much, and I'll pass it back over to Jan.
I want to start off by just actually, Dr. Winslow, can you speak a little bit more about the idea of CRISPR and gene editing in somatic cells, like you were talking about, versus the germline mutations that we've seen in the news, and just kind of touch on how your work is a little bit different. We don't need to go into the ethical aspects of it, but if you could just touch on that a little bit, I think there's a few questions about that.
Great. Thanks. Yeah, so there's obviously, a tremendous number of things that can be done with CRISPR genome editing, right? And what we hear a lot about is, the germline editing, right? Altering, organisms but what we're really thinking about here is really separate from that, right? We're really thinking about using this technology to understand, right, cancer. So like I gave the example where we have these cancer cell lines. Here's just an experiment that we can do within these cells to ask, what do they depend on? What do they need to keep growing? And then our work within these mouse models, ours and others, is really altering the genetics in adult cells within these organs where we want to understand cancer. So these are quite different. I would say another question that comes up quite often is, the thought that maybe we'll use CRISPR to cure cancer, right? If you can really delete any gene you want, you might think, well, why don't we just get CRISPR into the cancer cells and we can cure it. It ends up that this is not an easy feat. It's not easy to get these guide RNAs, for example, into every cancer cell. So really the two things I talked about today were understanding, right, which cells are required in cancers, and then understanding the genetics of these, how we drive cancer initiation and growth.
Great. A follow-up question to that is actually would this same approach work for diseases other than cancer?
Right. So cancer is, if you want to just think about the best application, cancer would be the best application, because cancer is a disease where mutations occur in adult cells, usually over time, and we can delete those genes or alter them in different ways and then look at cancer phenotypes. Many other diseases are perhaps driven by complex genetics, but they're entirely but it's more the genetics of the entire organism or the entire person, not necessarily that a gene was altered in an individual cell. There are probably other diseases like that, but in many cases, I think there's different approaches that could probably be used better in that regard. Sorry, I would say that I guess with that answer, I was a little bit stuck on thinking about the in vivo models that we use, right? But actually, there's many applications of CRISPR-Cas9 genome editing to understand biology around other diseases. Maybe taking different cell types like neurons or stem cells, and then altering them with CRISPR, and then asking questions about what different genes do. That's tremendously useful for understanding what drives a lot of different diseases, actually. So there's many different ways to think about this, but certainly these general approaches could be used to understand many different diseases. Thanks.
Great. You mentioned that the use of virus vectors to create an in vivo tumor growth. Can two different types of tumors combine so that they metastasize into a genotypically altered third tumor?
Oh, I see. Yes, right. If two tumors or I think mostly we would think about this slightly differently. I know in our models we generate a lot of individual tumors, like I said, maybe thousands of tumors. It's certainly the case that, for example, in human tumors, as they grow, there's different mutations in different cells, even within the growing tumor. So you end up with what we call subclones, kind of different parts of the tumor that have different genes altered. And it's certainly the case that different subclones could perhaps help each other and maybe even help each other metastasize. So if that's the question, then it's entirely reasonable that that would happen, but very poorly understood how there might be cooperation across different subclones. Hope I got that one right.
If we use a gene editing approach in cancer treatment, what are the challenges and limitations that there'll be, and what are concerns for patients?
Yeah, so the biggest challenge in using CRISPR for actual cancer therapy is that it's very difficult to get these required components into all cancer cells. So you can't just say, well, cancers need this gene and we're going to somehow get Cas9 and a guide RNA into every single cancer cell. It'll delete that gene and the cancer will go away. There's too many cancer cells. This sort of delivering those things is at this point not feasible. Now, you might imagine saying, well, what if there was a way to get some CRISPR-Cas9, guide RNA components into only 1% of cancer cells? And then for some reason, if you kill that 1%, now you have an impact on all the surrounding cells. Maybe you activate the immune system or, something like that. I think that's the way I would think about it more, which is saying, well, is there a clever way to not have to get these components into all the cancer cells? And then the second part of the question, I think, was sort of related to what's the risk to the patient. In a theoretical world where you could do this, I wouldn't see the risk is the same as any sort of cancer therapy, right? It's going to be side effects of having impact on normal cells. I don't think there'd be a tremendously different risk for this type of thing.
Okay. Now, a lot of questions are actually about the difference between CRISPR and using RNAi methods. And so people were wondering if, what's more specific in terms of, knockdown, what is better, what are the pros and cons, why is a CRISPR a better technology for what you're doing and being able to knock down the genes?
Right. Okay. So RNAi or RNA interference is a very different type of method where you express RNAs that bind to genes, actually the mRNAs of genes, and reduce the expression. So in that case, And then, of course, CRISPR is what I just talked about, where you have this Cas9 or other protein, other nuclease and guide RNA, and you actually cut the genome and you inactivate that gene. So they're very different for two reasons. One is that RNAi is transient, meaning that when your RNA molecule is there, it will reduce the expression of the gene, but over time it goes away. Whereas CRISPR-Cas9 genome editing, because you're actually altering the genome, is stable. So when you delete that gene, like we did in our models or in whatever system, it will always be deleted from those cells. So that's one big difference. Another difference is that when you delete genes, they're really gone. They went from there to zero. With RNAi, you're always knocking down the genes, meaning that you're reducing the expression, but probably not getting it to zero. So I think which method would be better kind of depends on what you want to do, right? So in some of our experimental settings, we really want to just inactivate that gene, have it be inactivated forever, and see what it does. There are different therapeutic applications where RNA interference is already being used, and even to essentially knock down genes and help with disease phenotypes. So they're very different approaches, and they have different pros and cons. Depending really on what you're trying to do. Yeah, it's a very interesting thing to think about, how best to do it experimentally, or if you're thinking therapeutically, how best to do it that way too. Thanks.
Oh, thank you. A couple other questions are asking about this idea of you're doing this everything in a mouse model. Mm-hmm. How does that relate to human? Would the next step be iPSC, stem cells, or would it be how? Mm-hmm. Yeah, can you just speak a little bit more about how mouse models relate to humans?
Right. Yeah, exactly. So, I think one key thing is what we start with is really human data. We start with an understanding of the genes that are frequently mutated in human tumors, in our case, human lung cancer. And then those are the alterations that we engineer in the mouse lung epithelial cells and then look for how they do or do not drive cancer. So at that level, the genetics were sort of matching. There's been studies to look at, for example, what does the tumor look like? If we take our tumors from the mouse model and we show them to our pathology colleagues here at Stanford, they'll tell us, oh yeah, it looks like a subset of human lung cancer. So that seems good. And we've done some other sort of analysis on mouse tumors that appear to align with human lung cancer. So there's several sort of analysis people have done that give us faith that these models are recapitulating at least aspects of human cancer. I do think it's really important also that these tumors in the mouse models are developing entirely, in the natural setting, right? From a single cell all the way perhaps to a metastatic cancer with all the normal sort of interactions. And this is a feature that, I do think the mouse is really quite similar to human, right? Lots of what we understand about disease biology, developmental biology has come from the mouse. And, so we do think about this, but I think it's a pretty actually parallel system to the human, but a system where we can control things, right? We can delete genes and we can ask what they do, and we can give therapies and, see what happens. So, yeah.
Okay, great. And then one last question. A few people were interested in how you're actually getting CRISPR and the guide RNAs into the cells. So it's a little bit more of a technical question, but if you could just expand on how that process happens in the lab.
Oh, yeah, right. So for the approaches that I showed where we have these mouse models of human cancer, we actually have the mice are engineered in a way where they have the ability to express Cas9 in any cell that we need it. So that's actually within the germline of the mouse, and there's these strains of mice that have the ability to do that. So Cas9 is within the mouse, and then the way we deliver the guide RNAs and other things that we want is through these viral vectors. So the reason I don't call them virus is, virus can replicate, and we all know that. These are essentially, engineered viral vectors, meaning they kind of are like a virus and they can deliver something to a cell, but they can never replicate. So people use this a lot experimentally to essentially get things into cells, and we use this type of virus called the lentivirus, but essentially just as a vector to take what we want and then get it into the cell. So that's how we do it. I would say across many different systems. There's lots of different ways to do it. So, for example, we all know that you can package mRNA in lipid nanoparticles now, and you can deliver it to cells. That's how the Pfizer and Moderna vaccines work, right? So there are different ways to get what you want into cells, and then, yep, exactly. It's slightly different in vivo and in cell culture, but there's lots of methods for that. It probably depends what you want to do. Great, thanks.
No, thank you. And if you would like to learn more from Dr. Winslow and the rest of the genetics faculty, we encourage you to join our genetics and genomics program. You'll have more than 90 hours of content to learn from, and as well as access to videos, exercises, and other types of activities that will really help you sharpen your skills. Once again, thank you for joining us today. We hope you have a great weekend, and we hope to see you again in future sessions.